today's session we will focus attention onto the understanding of proteins, and we will have prime focus will be on the classification and structural organization of proteins. The session is planned as follows. To begin with, we will introduce ourselves to what are proteins. Thereafter, we will take up the classification of proteins. We will use different bases for their classification, and then thereafter we will come down to the structural organization of proteins. In this context, we will talk about different levels of organization of protein structure, and then towards the end we will try to touch upon protein denaturation, what are the factors causing denaturation, and lastly we will try to sum up what we do in today's session. Okay. Now, let us make a beginning. Let us ask ourselves what are proteins. I am just trying to define it for sake of it, because most of us know what proteins are, but just for sake of it, let me put into words. Basically, proteins are a group of highly complex organic compounds which are found in all living cells, and they comprise the most abundant class of biological molecules. What it means is that they are the molecules which are present in the body or any living being in a very large amount, to the extent that in the human body about more than half the dry weight of the body is made up of proteins. It is such a important biomolecule for us, and more so proteins perform probably the widest range of functions in the body. We will talk something about it as we proceed. Further, in terms of putting it in terms of conceptually speaking, or in terms of chemistry, according to IUPAC, proteins basically are defined as naturally occurring and synthetic polypeptides having a molecular weights greater than about 10,000 units, 10,000 Dalton as biochemists call it. So, you will recall that we did mention something of this kind when we talked about peptides, because when we tried to classify peptides, we talked about polypeptides, and then we kind of distinguish between polypeptide and protein. When a polypeptide becomes very large, we start calling it as protein. So, with this line of demarcation between polypeptide and protein is just arbitrary. It becomes larger, we call it as protein. Okay. Now, this is about the part how do we look at proteins. This term protein basically was coined by Johannes Mulder in 1839 to define a class of biological molecules, because at that time people were trying to look at biological molecules and the proteins were isolated, and the name was given on the basis of Proteus. It is an early Greek sea god whose name Proteus means basically first or primordial or of first importance. Basically, the underlying principle here is that the Mulder wanted to emphasize the significance, the importance of proteins. That means they are the most important biological molecules in the system. Now, let us move on to how do we classify proteins. The number of proteins available is enormous, very large amount. Why large number is there? And then there are different ways of classifying them. Some of the bases on which the proteins are typically classified happen to be the number of polymeric units, that is one. Another one is the chemical composition. A third one happens to be the biological function, and another one is about the shape of it, how do they look like. Let us take each one of these classification one by one and understand what do they mean to us. Let us look at the classification based on number of polymeric units. On the basis of number of polymeric units, one typical way would be that we have got proteins which are monomeric. As the name suggests, mono means one. A protein molecule in which there is only one peptide chain is present, because you may have proteins where there are more than one units are there, we will come to that. So, monomeric proteins are the ones in which they have only one long peptide or polypeptide chain. The second class happens to be multimeric. Obviously, a multimeric is a protein in which more than one peptide chain is present over there. In such cases, these peptide chains which constitute the protein as a whole, they are referred to as protein subunits, and these protein subunits may be identical, may be different, because you may have, like hemoglobin happens to be a multimeric protein. So, there are 4 subunits in that, 2 of one kind, 2 of another kind. So, that is the meaning of the molecule to be multimeric. Let us take another example, insulin. Insulin also is an example of a multimeric protein, because in this case there are 2 protein subunits, there are 2 chains in that, and the 2 chains happen to be different. So, that is one way of looking at the classification based on the number of polymeric units. Another way of looking at classification is on the basis of their chemical composition. If you look at the chemical composition, proteins probably can be put into two broad classes. One happens to be the simple proteins. Simple proteins are the ones basically they consist only of amino acid residues. That means you have a long chain which consists of only amino acids, nothing else. There is no kind of a compound or other component present in the protein. Whereas the second class happens to be complex proteins, or also called as conjugated proteins. Now, these proteins are the ones which contain one or more non-amino acid entities in the structure, in addition to one or more peptide chains. What it means is the complex protein, as the name suggests, is very complex. It may have maybe 1, 2 or more than 2 protein subunits, and also there could be some component which is in addition to, which is non-amino component. There is a non-amino acid component over there. So, that is a conjugated protein. Now, this in the conjugated protein, that the non-amino acid component is termed as a prosthetic group, and this prosthetic group may be organic in nature or inorganic in nature. Maybe it is a small maybe a coenzyme is there, or maybe a metal ion is there, or maybe a carbohydrate unit is there. So, there are different kind of prosthetic groups which are possible in such groups there. Further, in such a kind of a conjugated protein, the protein part of the conjugated protein is called as apoprotein. The non-protein part is called as prosthetic group, we have such seen, and the entire molecule, that is a combination of apoprotein and prosthetic group put together, is referred to as haloprotein. That is just for sake of it, I am just trying to put terms to you so that we can keep in mind. Now, let us move further. Let us move further and see that these conjugated proteins, as I mentioned, is a very vast class, because there can be wide variety of things which get conjugated to the protein molecules to give complex or conjugated proteins. And based on what is the prosthetic group in a given conjugated protein, conjugated proteins can be further classified. Like you have phosphoproteins. As the name suggests, in this case the prosthetic group happens to be phosphoric acid. Yet another one could be metalloproteins. So, in this case the prosthetic group or the non-protein part happens to be some kind of metal ions over there. Yet another could be chromoproteins. Chromoproteins basically are the proteins which contain some kind of colored pigment as a non-protein component. Examples happen to be carotenoids as the pigments over there. Then you may have glycoprotein. As the name suggests, glyco, basically that means they have non-protein component to be carbohydrates in nature. So, and so on and so forth. There are large number of classes in which the conjugated proteins can be further subdivided. We will not get into all the classes, I just mentioned a few of them for your reference. Okay, let us move a little further and see how do we classify proteins on the basis of the biological function they conduct, because we did make a mention here that proteins probably are the most important biological molecules, because they perform widest possible range of biological functions, and just for sake of it we categorize them, and on the basis of the biological function the proteins could be classified as follows. That is, one could be the structural function, because this is one of the most important function of proteins there. As the name suggests, these provide mechanical strength to large animals, because we have got muscles, the muscular proteins there, or keratin, I have given an example of that is a hair protein, a protein in the nails and all. So, they are the ones which provide structure to the body. So, they are called as structural proteins, because they provide mechanical strength, support. The second class happens to be regulatory proteins. This again is a very important class here, because these proteins will control different aspects of cellular function. For example, insulin happens to be a hormone. So, insulin also is a protein which is acting as an hormone. So, that is a kind of a regulatory protein, because hormones have a regulatory function to perform. Then another function of proteins happens to be protective, because these are called as protective proteins. As the name suggests, these proteins basically defend the body against infection. So, say maybe there are certain animals have toxins which belong to this category, or maybe you can have immunoglobulins, the things which kind of protect us from the infection over there. So, they are called as protective proteins. Then you have catalytic proteins or enzymes as they are commonly called. So, these catalyze the synthesis and other kind of reactions in the body. One example happens to be dehydrogenase. That is a common enzyme we know of. Then you can have even transport proteins. As again, you can get the function from the name itself, transport. That means these are the proteins which bind and carry specific molecules or ions from one place to another in the body. The most common example happens to be hemoglobin. We know that hemoglobin is the one which carries oxygen in our body, or even carbon dioxide. So that is a transport protein. Then you can have contractile protein. These are the proteins which provide cell and organisms with the ability to move, change shape. Myoglobin happens to be one of the examples of that. That means the muscles movement over, this is done by myoglobin there. So you see that the proteins are important in wide range of activities in the human body or any other living organism for that matter. Okay. Now continuing with this, yet another function which the proteins perform in the body happens to be the storage protein. So these act as a reservoir of nitrogen and other nutrients which can be used when the external sources of are either low or they are absent. For example, albuminin, that happens to be your egg protein, because we do consume eggs as a source of protein, or maybe some of the milk proteins are there. So they are the proteins which can be, because we know that proteins are the most important reservoir or storage for nitrogen and other nutrients. Suppose the body is lacking in food, then what happens? Suppose I am on fasting, my body is not consuming any food, but I do need energy to survive. So what body does is, after consuming all carbohydrates, the reserves, it starts attacking the proteins part. It breaks down the proteins and gets energy from there. So that is the kind of function we have for proteins. Okay, let's move a little further and talk about classification on the basis of the shape the proteins occupy or acquire. On the basis of shape, there are proteins probably can be put into two main types. That happens to be fibrous proteins. As the name suggests, fiber, long, elongated ones. So these are proteins whose molecule have an elongated shape. That means one dimension happens to be larger than another one. So this example here, this is the example of collagen. Collagen is a protein which is a fibrous in nature. You can see that it is elongated. That means the length of the molecule is much larger than the width of it. And this ratio, length to the width, is referred in terms of axial ratio. And for fibrous proteins, this happens to be greater than 10. That means length is much larger than the width. So these tend to have very simple, regular, linear structures. Examples are keratin, collagen. The example of collagen is shown over here. So that is the kind of proteins we have. The large number of proteins which belong to this category are of fibrous proteins. They are long, elongated proteins there. The second category is called as globular proteins, as the name suggests, globular, globule like. So these are proteins wherein the peptide chains fold into some kind of a spherical or a globular shape. So that is how they look like. That means the protein molecule is not elongated one, rather it folds and acquires a globule kind of a shape. That is why the name globular proteins. Now in this kind of a proteins, what happens is the amino acids with the hydrophobic side chains are in the interior, and the most of the hydrophilic side chains are on the outside. You remember when we talked about amino acids, we did talk about the side chains there. So some side chains are hydrophobic, the aliphatic amino acids or the apolar amino acids. The chains are hydrophobic. So when the proteins fold, all such chain will get kind of into the core of the protein. They will not expose themselves because outside is water, and that is why. So the protein surface will be covered by the amino acids whose side chain are hydrophilic in nature, because they will try to interact positively with water, whereas the hydrophobic chains will try to go away from water and get embedded into the folds of protein there. So that is a kind of shape and structure we have for the globular proteins. Now these proteins are water soluble substances and usually function as enzymes, hormones or transport proteins. So that is a kind of behavior they have or the kind of functions they perform in the body. Okay. Having talked about the classification of proteins, let us now move on to the structural organization of proteins, because proteins come in wide variety of shapes and structures. In terms of organization, so typically there are 4 different levels of structural organization. They are referred to as the primary structure. That is a first level of organization of protein structure. The next is the secondary structure. The third one happens to be tertiary, and the fourth one is quaternary structure. So, that is how hierarchy it goes: primary, secondary, tertiary and quaternary structures. We will talk about each level of structural organization and understand the meaning and significance of each one of them. Okay, let us begin with the primary structure. The primary structure of proteins basically refers to the sequence of amino acid residues. We learnt about amino acid residues in the previous sessions there. Now, what does it mean? What does it mean is that if I am talking about a protein, because every protein has a very characteristic or a specific sequence of amino acids in that, and that particular sequence refers to or is called as the primary structure of protein. Let us see that. And also, as we know that the proteins are synthesized in the body on the basis of the blueprint carried by the genetic molecule, that is DNA. So, the genetic information in DNA is transferred into synthesizing the protein there. So, the sequence, what are going to be the amino acids in a protein and in what sequence are they going to be there, that all is controlled by the genetic code of that. So, the sequence, the primary structure of protein is governed by the genetic information on DNA. The next point which is important to us is that, let me take an example here. This picture here shows, as I mentioned earlier, insulin molecule has got two chains there. One of them happens called as A chain here. So, what I am showing you is the sequence of A chain. So, what you see is that in this A chain there are about 21 amino acids there, and they are in this particular sequence over here. That means, this is my C terminal. So, from this C terminal, as I move, we have asparagine here, cysteine, tyrosine, asparagine and so on and so forth. We continue like this and we come down to our N terminal, which happens to be glycine. So, otherwise typically, as I mentioned earlier also, that when you talk about proteins, we start from N terminal to C terminal. The sequence will be, say, glycine, isoleucine, valine, glutamic acid, glutamine and so on and so forth. So, that is what I am referring to. When I say I am talking about insulin, I must specify that what are the amino acids, in what sequence are they there, because if I change any of the amino acid, this will no longer be the A chain of insulin, because that will not be insulin molecule at all. It will not be able to perform the function it performs, that is acting as an hormone. Further, another very important thing here is that this primary structure of protein is the one which determines the other orders of structure, because primary structure is going to determine what happens, what kind of secondary structure does it adopt, and secondary structures in turn will lead to the tertiary structure, and thereafter we talk about the quaternary structure. So, this we move in a hierarchical order from primary to secondary to tertiary to quaternary structure, and the overall shape of the protein and the function it performs is controlled or is governed by the primary sequence or the primary structure of protein. So, it is of the prime importance. This is the most crucial component of any structural organization of protein there. Let me take some examples here. I give you examples of two peptides. I am not talking about proteins, but the argument remains the same. Look at these two peptides here. The first one is oxytocin. It is a nonapeptide. That means there are 9 amino acids in that, and they are in this particular sequence over here. And a very closely related peptide happens to be vasopressin. This also is a nonapeptide, but these two peptides have got roughly similar shape and similar sequences also, but there are only 2 points of differentiation here. Say this point of differentiation is that in case of oxytocin at number 3 we have got isoleucine, whereas in case of vasopressin I have got phenylalanine here, and at position number 8 here we got leucine and here we got arginine. So, there are 2 points of differentiation. That means, this is oxytocin, and in oxytocin if I replace this amino acid residue by this and this one by this one, respectively change this one, I find the 2 peptides are very different there. Oxytocin is the one which regulates uterine contraction and controls lactation there, whereas vasopressin again is a peptide which regulates the excretion of water by the kidneys, entirely different function. We just see that by changing these 2 amino acids from one peptide to another one, we have got a entirely new peptide with entirely new function and the behavior. Okay, so, that is just to emphasize the importance of the primary sequence there, primary structure of a protein or an amino acid, protein or a peptide. Similarly, if you look at the structure of hemoglobin, even in hemoglobin, you know that hemoglobin is a transport protein which carries oxygen in the body. But if आई थिंक इसको छोड़ देंगे, हीमोग्लोबिन पार्ट छोड़ दीजिएगा, जितना ये है उतना ठीक है, क्योंकि मुझे उसका याद नहीं आ रहा कि कौन सा चेंज है, उसको छोड़ देते हैं, जितना यहाँ बोला गया वो ठीक है, मैं आगे कंटिन्यू कर रहा हूँ. ओके, नाउ लेट्स मूव ऑन टू द सेकेंडरी स्ट्रक्चर. Let's see what do we understand by secondary structure of proteins. Secondary structure of proteins, I'm just trying to quote to you according to what is the definition according to IUPAC, IUB, that is International Union of Biochemists. So, they have defined secondary structure of proteins as follows. We have to understand, this is very, very crucial for us. That means the secondary structure of a sequence of a segment of a polypeptide chain is, now mark my words here, that is the secondary structure of a sequence of a segment of a polypeptide chain. That means I am not talking about the secondary structure of a protein as a whole. Protein is a very large molecule, we may have hundreds of amino acids in that. Out of these large components, small segment I am referring to. So, what is the structure acquired by this segment? So, that is the secondary structure, first part of it. Secondly, so it is a structure of a sequence of segment of polypeptide chain is the local spatial arrangement of its main chain atoms without regard to the conformation of its side chains. That is one part. That means I am referring to firstly out of the whole protein molecule, small segment of that. Within this segment, I am not talking about the whole molecule, I am just looking at the structure, the three-dimensional structure acquired by the backbone main chain atoms. Main chain atoms are our C alpha, CONH, C alpha, CONH. So, that is a backbone I am talking about. So, the polypeptide backbone, what is the shape that acquires? And without regard to the conformation of its side chains, we do not bother about, when we talk about the secondary structure, we are not concerned about what is the kind of conformation of the side chains. Or suppose the protein happens to be of more than one subunit there, then we are not bothered about what are the position or the spatial arrangements of other segments there. So, what it boils down to is that when we talk about secondary structure, I am primarily referring to out of the whole large protein, small part of it, and there again I am just referring to the spatial arrangement of the backbone. That is what it means. And there are two types of structures we have here, and the structure happens to be alpha helix and the beta sheet. Let us understand each one of them. Alpha helix first. Now, alpha helix, this picture is very familiar to all of us. This structure was proposed by Linus Pauling, the Nobel laureate who got Nobel Prize twice, once for chemistry, one for Peace Prize there. Now, this was proposed by Linus Pauling and Corey in 1950. Basically, what they used was, they used the dimensions of peptide bonds, which we learnt about in the previous sessions, and the kind of steric considerations, because we know that peptide bond is a planar peptide bond. So, we have got backbone CH alpha, CONH, CH alpha. So, that protein, that kind of backbone has to be there. So, it cannot move within the peptide part of it, only the other components can move. So, with that kind of constraints, where can it move, what kind of structure can it require? So, it was proposed by Linus Pauling and Corey that this kind of a structure is possible, wherein the peptide backbone acquires some kind of a helical structure, that means like a screw kind of a structure, and that such a structure has to be stabilized, and this kind of structure is stabilized by hydrogen bonds. We will talk more about it as we move further. Now, basically this is a kind of a coiled structure which is stabilized by hydrogen bonds, as I just mentioned. Now, let us look at some of the salient features of an alpha helix. Firstly, now the oxygen atom of every carbonyl group of the peptide bond forms a hydrogen bond with the hydrogen atom of an amide group in the next turn of the helix. Let us understand that first. Let us first understand what do we mean by a helix. The protein alpha helix is a right-handed helix. What does it mean? It means the following. That means, if I move along the backbone of the protein, if I start from here, say this is my end terminal, if I start moving along the backbone, as I move like this. That is, as I move along the backbone, I move forward. It is like a right-handed screw. What do we do in a screw? We take a screw, turn it clockwise. As I turn it clockwise, it moves forward. same thing happens here. That means, as I move along the backbone of the protein, I am moving forward. So, that is the meaning of it being a right-handed helix. Secondly, when you have got this kind of a thing, so what you have is the peptide backbone, I am repeating again, is essentially CH alpha, CONH, CH alpha, CONH. So, that is what is moving like this. So the first amino acid here, it has a CONH. So, then as I move further, the protein will take a turn and come back. So, another peptide bond will be there. So peptide bond which is here, the carbonyl group here, when it moves down, there is a NH there. So, this CO and this NH, they form a hydrogen bond. So, as I move, the next peptide bond CO here, NH will come there. So, that is how it happens. That means, you have got a helical ordered arrangement there, wherein each carbonyl gets into a hydrogen bond with a NH group as it moves along further. So, it moves, takes one turn and then forms a hydrogen bond there. So, that is the kind of arrangement we have in case of alpha helix. So, as I mentioned, it is a right-handed helix. Then the hydrogen bonds of alpha helix are parallel to the long axis. So, that means, as I just said, it is a axis like this, we are moving a coil. So, hydrogen bonds are running parallel here. So, this is axis here. So, this is my helical axis there, this is my axis, the upper direction there. So, these are my hydrogen bonds which are running parallel to that. That is characteristic of alpha helix. Yet another one is the repeat distance of the helix or its pitch is about 0.454 nanometer, and there are about 3.6 amino acids per turn of the helix. What does it mean is the following. That means, if I start from one end of the helix, I move, I take a turn and come back. So, this is my one turn. So, when I make one turn, I travel a distance of about 0.54 nanometers, and in doing so, there are about 3.6 amino acids are used in this process. So, that is the meaning of this particular statement here. Further, the R groups of the different amino acids extend to the outside of the helix, because our hydrogen bonds are running parallel. You can see that the R groups are just on the periphery, they are coming out of the helix there. And mind you, our secondary structure refers to only the backbone part of it. We are not concerned about the R groups there, the side chains there. Okay. Now, let us look at another secondary structure possible there, which again was proposed by Linus Pauling and Corey, and this is referred to as the beta sheet structure. Beta sheet structure is a bigger structure, but the basic unit for this structure happens to be a beta strand. Basically, a beta strand refers to a fully extended polypeptide backbone chain. That means, we have got a polypeptide chain, we do not make it helical, let it just get extended there. So, we got a long elongated chain there, that is called as a beta strand. Now, due to the rigid peptide bond structure, so this amino acid, when you just stretch it out, we find that the amino acid side chains will alternate. So, as I move further, so one R will be here, another will be here, then another here, and they will alternate like that. Then these long elongated strands do not stay as such, they do not stay stiff like this, but what happens is, you have a peptide backbone moving straight, then it takes a turn, comes back, then turns back like this. So, when it does like this, it forms a sheet kind of a structure. That is how it looks like. That means, you have got an extended polypeptide backbone here, and then this peptide backbone moves, takes a turn, comes back, moves like this, again turn and comes back. It forms a sheet kind of a structure. When it forms a sheet kind of a structure, we find that wherever there is a CONH group there, so the carbonyl groups here, this is my carbonyl O here, this will be NH here, so they will form a hydrogen bond there. So, what happens is, one strand of this will have CO groups there. So, when the other strand comes, there will be NH there, they will form a hydrogen bond there. So, in this case, again the structure is stabilized by hydrogen bonds, but in this case the hydrogen bonds are not parallel to the axis. In this case, they happen to be perpendicular, because this is my sheet here, my hydrogen bonds are perpendicular to that. So, that is one differentiation between two kinds of structures. Further, there are basically two arrangements possible here, because what happens is, when this kind of sheets are formed, you may have situation that you may have an antiparallel beta sheet arrangement, wherein as the sheets move, we find that N termini of one side, they are aligned with the C termini. This is basically an antiparallel kind of arrangement here. And another possibility could be the two chains, when they fold and make a sheet, they are parallel. That means, the N terminal here and N terminal here, they will kind of align with each other. So, same will be true of the C terminals there. So, there are two arrangements possible there. there is yet more aspect of this structure, that is because the side chains, because we have extended chain there which is forming a sheet, fine, but there are side chains are there which are bulky, they may be very bulky and they do not let it remain flat. So, what happens is this sheet acquires some kind of a pleated kind of a structure there, that is why it is called, they had some kind of a twist there. So, they are called as beta pleated sheets, and such a structure is referred to as beta pleated sheet, because this pleating is coming because of the bulk of the R groups over there. Now, when we refer to higher order structures, say tertiary structure and further on, so such segments, the secondary structures are normally shown in terms of some kind of a shorthand symbols there. Typically, these two structures like an alpha helix we do not draw the whole structure, we just show a coil of this kind, a flattened coil. This basically refers to an alpha helix, and an arrow of this kind, a flat arrow is referred to as, is indicative of a beta sheet structure. When we talk about, we will just see in a short while from now, when you look at a tertiary structure, that is how they typically are shown. Okay. It is time that now we move on to the tertiary structures. The tertiary structure of protein, once again we repose our faith into the definition from IUPAC. So, according to IUPAC, the tertiary structure of a protein molecule or a subunit of a protein molecule. Now, again, when you talked about secondary structure, we were referring to some component of the protein molecule. Now, I am referring to protein molecule as a whole. That means, one molecule of protein or one subunit of a large protein when there are more than one subunits are there. So, that is what it means. That means, I am talking about a still higher order there. I am talking about a protein molecule as a whole. Further, it is the arrangement of all its atoms in the space without regard to its relationship with neighboring molecules or subunits. That means, I am referring to the structure of one protein molecule as a whole, irrespective of what is the structure of the next subunit over there. So, I am talking about the structure of one full protein here. In simple words, this basically refers to the characteristic three-dimensional structure of a given protein molecule. Such a structure is referred to as native structure or native structure of the protein. We will talk about it when we talk about denaturation there. Now, how do we schematically represent? This is one picture which I could just lay my hands on. This just gives you an indication of how do we refer to a tertiary structure. We can see that this is one protein molecule, a large protein molecule, and what you see is that, so I start from here. This protein molecule has, there is no structure part of it here. I mean, this is just some kind of a random structure here, or random coil as it is called typically. Then this component happens to be a beta sheet, because that is a flat arrow is there. Then another beta sheet, and we keep moving along the backbone, another loop is there, and then we find there is an alpha helix there. So, then another random kind of structure, we can have alpha helix. So, that means that tertiary structure basically refers to sum total of the structure of a protein molecule inclusive of all its components over there. That means, if I just look at a large protein molecule, maybe suppose I am talking about a 500 amino acid protein. So, maybe first 50 amino acids do not let any structure come over there, next 72 make it alpha helical, then maybe some random component, then there is a beta sheet there. So, all put together, what is the overall structure? That is the meaning of a tertiary structure of a protein. Now, when we talked about the secondary structure, we talked about alpha helix and beta sheet, we realized that it is a hydrogen bond which primarily is responsible for the stability of the structure. In case of tertiary structure, there are many more forces involved. Let us see. So, in this case of tertiary structure, a range of interactions contribute to stability. Now, they happen to be, say, could be hydrophilic interaction. Maybe on the periphery here there is one side chain which is hydrophilic, so is another one. So, these two can interact hydrophilically, or maybe in the inner pocket here you can have a hydrophobic interaction. Hydrophobic interaction basically is the interaction between two side chains, both of which are aliphatic in nature. That means, say this side chain here, this is phenylalanine, this does not like water, it puts the water away, so does this one, the two come closer. So, such an interaction where two groups which do not like water, they like each other. This interaction is referred to as hydrophobic interaction. Or another possibility could be, say, disulfide bonds. You remember that when we talked about amino acids, we talked about cysteine, and we said, we made a mention there, the cysteine is very unique in itself, that it can form disulfide bonds. and this has a very significant role to play in stabilizing the structure of protein here. We see that this is a disulfide bond here. That means, on this protein here, somewhere here there is a cysteine amino acid residue there. When you move further around there, another cysteine here, the two can get into a covalent linkage. It is a very strong bond here which keeps it protein into this particular shape. Or maybe we can have some kind of hydrogen bonds over there, or maybe we can have salt linkage. Salt linkage basically refers to, say, maybe on this part of the protein there is a say lysine side chain is there, which has a amino group as the side chain there, or we can have a glutamic acid or aspartic acid with COH group there. So, they can form acid base interaction, some kind of a salt linkage. So, wide variety of interactions come into play to stabilize the tertiary structure of a protein molecule. Okay. Let us move further and talk about the quaternary structure of protein. Again, we go back to IUPAC and see the quaternary structure of a protein molecule is the arrangement of its subunits in space. That means, now we are talking about more than one protein molecule. That means, I am talking about the arrangement of the subunits of a larger protein, small components of that, how does that subunit behave, and the ensemble of its inter-subunit contacts and the interactions without regard to internal geometry of the subunits. We are not bothered about what is the internal component, we are just talking about how are the subunits arranged, that is the quaternary structure. And the most typical example to quote here is that of hemoglobin. In case of hemoglobin, there are 4 subunits there. So, typically they are of 2 types, there are 2 are alpha chains, 2 are beta chains, but in this picture here they are shown to be of different color just to indicate that there are 4 subunits there. So, when I am referring to the quaternary structure, so when I talk about hemoglobin, I refer to the hemoglobin, the subunits arranged in a particular orientation or a particular arrangement, that is the quaternary structure of this particular molecule. Now, having talked about the protein structural organization, let us see learn something about what do we understand by protein denaturation. Protein denaturation basically refers to the destruction of native 3-dimensional structure of the protein by different agents. There are a number of agents which can cause this disruption and causing it denaturation. Now, agents could be simply as heat. Suppose I look at supply some heat to a protein molecule, this heat will provide energy, and that energy will be sufficient enough to break the hydrogen bonds. When the hydrogen bonds are broken, we see that the secondary structure is lost, so is that even some parts of the tertiary structure also is lost. So, as a consequence, the protein loses its structure and function, and typical example happens to be coagulation of egg white, because we know that egg white is fluid like, but if I make the egg boil, when I boil it, it becomes hardened there. So, that is just because of the denaturation of protein. Similarly, we can have variety of agents over there like strong acids, UV radiation, solvents there, or mechanical, even agitation can also cause protein denaturation, or we can have metal ion poisoning and all things. There are variety of things over there which can cause denaturation of protein. Okay. Now, let us sum up what we have done today. What we have done today is we started by just looking at what do we understand by proteins, then we primarily spent a lot of time on the classification of proteins. Thereafter, we looked into in detail on the structural organization, wherein we talked about the primary, secondary, tertiary and quaternary structure of proteins. And lastly, we just touched upon the meaning of protein denaturation, and then we mentioned the number of agents which can cause denaturation of protein.